Hello, welcome ladies and gentlemen to today's tutorial. Today we are going to look at muscle tissue. Muscle will actually constitute about 40% of the total body weight, yes, in humans. Yes, with regards to this tutorial, we're actually going to look at, the muscle tissue. We're going to look at the functions of muscles. We're going to look at actually the properties of muscles. We look at, yes, the organization, how the muscle is organized, yes, from the gross aspect to the microscopic aspect, even to the molecular aspect. Then we actually end with actually knowing how we can identify the various types of muscles under histological sections. So let's begin our journey looking at this muscle tissue. So starting with this, we want to actually understand some key concepts, regarding the study of muscle tissue. So the first concept that I want to have, bulleted over there is myology. Myology is the study of, muscle. Logy has to do with study, and myo has to do with muscle. Yeah, there is another prefix that also, explains what, this muscle, tissue is, or what muscle is. And whenever you hear the word sarco, has to do with muscle. So, for instance, we hear of, sarcolemma, which is the cell membrane of the muscle cell. Now, the other thing is that we have to, actually understand, some functions regarding, the muscle. So importantly, the muscle which will get, surfaces to attach on the bone. We've already explained that the skeletal system is going to aid in movement. By so doing, it will provide surfaces for attachment to these muscles. So for instance, to the skeletal muscles. So that upon contraction and of course, relaxation, it will cause, movement. So the muscles will be very relevant in movement. Yes, they will also be relevant in actually what we call, stability, as well as, posture. Yes, they will also be relevant in what we call, play a key role in the circulatory system, the cardiovascular system. For instance, the contractions of the muscles of the heart will cause, blood to be ejected, to various parts of the body, Apart from that, even in venous return, what will happen is that yes, we have what we call, for instance, the soleus, in the deep, veins in the lower limb, especially in the leg. Upon contraction of the soleus, it will help in what we call venous return. Soleus is actually muscle, which is located in the posterior compartment of actually the leg. So even apart from that, if we look at the tunica media of blood vessels, is made of smooth muscles, which of course upon contraction will help in either vasoconstriction or vasodilation, depending on the needs of the body. Yes, apart from these one, even in the respiratory system, it is going to play an important role. Yes, regarding even contraction and relaxations of the diaphragm. The diaphragm is actually a muscular sheath, which is going to be very important in breathing mechanisms. Apart from that, the antagonistic contraction and relaxations of the external intercostal, and of course internal intercostal muscles will actually help in various movements, yes, to actually either increase the volume or decrease the volume of the thoracic, or cage, thorax actually. So actually the skeleton, that will be important. These muscles are going to be very important regarding the respiratory system. Apart from the respiratory system, they will also be relevant in the digestive system. For instance, the muscularis mucosae, the smooth muscle over there, yes, even the external proper muscle, muscularis propria, yes, they are going to actually perform, various contraction and relaxation movement to either bring, for instance, the smooth muscles, the muscularis mucosae, will contract to bring the luminal content, that's the food product within the lumen of the digestive tract or the alimentary canal closer to the mucosa, so that various secretions can be put on the food. And apart from that, even various segmental movement and peristaltic movement will be aided by these muscles over there in the digestive tract. Generally, yes, these muscles will also contract. They were actually taken at some point to form what we call sphincters to guard the entrance of various openings of the body. Therefore, allowing some particles to move or controlling the movement of content from one region of part of the body into another. Yes, even the urinary system, yes, in the of course digestive system again. Sphincters are going to guard, as we've said, even yes in defecation, in urination, maturation. Yes, all these things are going to help control all these activities and biochemistry of these muscles. Even in childbirth, yes, where lady when a female is in labor, yes, under the influence of oxytocin, then of course there will be uterine contractions to cause the fetus as well as the placenta to expel out of the womb. Yes, even apart from this, an important thing is that yes, even in vision, yes, for you to change the position of your eyeball to have various eye movement, then it's very important that the extraocular muscles, yes, will have to actually move very well, yes, and that is very important, so that if in situations where the extraocular muscles are not able to contract and relax properly and cause different movement of the eyeball, then of course it can lead to what we call squint or strabismus. So these ones can happen. Even the sphincter pupillae and the dilator pupillae, yes, depending on the quantity of light present or the light intensity present, then it can actually cause constriction or dilatation of the pupil. So all these things are very important regarding the functions that muscles will play. Yes, even in the abdomen, although yes we don't have that kind of bony protection, but these muscles are able, of course, rectus abdominis, external oblique, internal oblique, transversus abdominis muscles are able to confer some bit of partial protection to the abdominal organs or the abdominal viscera. Yeah, it's very important. In the face, they were very important in producing various movements of the face for various non-verbal communications. Very important. Even in mastication, yes, chewing of food, yes. The masticatory muscles will play important role. So actually, the muscle is going to play several important roles. But finally, what I want to add is the shivering thermogenesis. Shivering thermogenesis. I've already mentioned non-shivering thermogenesis, which will be by the candidacy of brown adipose tissue, the multilocular, adipocytes who are going to do that. But for the shivering type, in situations where perhaps you are feeling cold, then there should be the need for shivering thermogenesis, where your muscles will will contract, involuntarily, yes, to cause heat production in the body. So these are some bit of the functions that these muscles are going to play. Now these muscles are able to play these functions because of some inherent properties that they possess. So the properties that they possess, yes, I've used the mnemonic. There are four main properties of the muscle. And these four main properties are the CE3. And C is contractility. So muscles have the ability to contract upon being stimulated appropriately. And therefore, it tells us that they will also be actually excitable. That's the E. They will be excitable, or they will be irritable, so that they can respond to stimuli. That's the meaning. And then again, they are also elastic, so that of course they can actually, upon contraction, then they can return to their normal position. And even apart from this, yes, they are also going to be extensible, so they can actually contract, or they can actually relax or extend stability, so they can extend beyond their normal limits. And at the end of the day, as they are elastic, they will be able to return back to their original position, original length. So generally that's what we are going to see. Now usually as we go through see the various types of muscle cells that we are going to see, We're going to see the skeletal muscle cells, which are usually attached to bones. Then, of course, we see the smooth muscles, which are also called, the visceral muscles, which will lie in various internal, structures. Then, of course, we also see the cardiac muscle, which are, mainly you're going to find it, in the heart. But we also learn some other areas, which, of course, which may be, having some, cardiac, muscles as well. Now, usually, if the muscle cell is stressed, okay, due to, of course, its high demand in function. For instance, if you are raising a lot of weight, then there will be the need for you to respond to the increased stress or the increased demand for you to work. And it can either go through what we call hypertrophy or hyperplasia. Usually, yes, if it is skeletal muscle, it will go through what we call hypertrophy. Skeletal muscles are usually quiescent, so that they are usually in the G0 state. Their cells are in the G0 state, so that they are non-mitotic. They won't be able to go through mitosis. But recently, some studies are finding that yes, even the skeletal muscle can regenerate. But generally, yes, they are not able to regenerate. But rather, what they will do is that if there's increased stress, then the cells will actually increase in size, go through what we call hypertrophy. So increase in size. Yes, even cardiac muscles will go through some bit of hypertrophy. Yes, so that if there is increased demand, if there's too much blood pressure, too much narrowing of blood vessels, then of course the heart will have to pump the blood very to the very great extent, and it will do so by actually going through hypertrophy, so that it can powerfully contract, and that one will usually lead to cardiomegaly. Then of course for smooth muscles, yes, usually they would respond to stress by what we call going through what we call hyperplasia. So that of course if, for instance, a lady or a female conceives, then as a embryo, and of course the fetus increases in size, then of course there's stress on the womb, and therefore the cells of the womb, yes, they will go through what we call hyperplasia, so they will increase their number. So these cells, yes, they are very stable. They will actually enter the G1 stage of the cell cycle, and then of course go to the S, and of course G2, and of course go to the M stage to go to mitosis, and of course give rise to new cells. So smooth muscles have high regeneration ability, So that is one thing that we are going to find. And then for atrophy, if the muscle is not put to use, then of course the muscle will go through atrophy, so that it will actually shrink, so to speak, and of course it decrease in size. And when that happens, then its functional ability will also go down. Now, usually associated with these muscle cells are another group of cells. We call them satellite cells. Now, these are not satellite cells which are in the the nervous tissue. Now these are which are the neurogland. We are not talking about that one. These are cells which are associated with the muscle cells. And of course, in situations where there is the need for the muscle cell to actually enter the cell cycle, G1 stage, and go through it, it will be converted, it will differentiate to become what you call myoblasts. Myo has to do with muscle, and that's the stem cell of the muscle to actually go through, and then eventually, so that stem cell is going to be the satellite cell, which will differentiate to become myoblasts, and then eventually form the muscle fibers. Now I also explain what we call the motor unit to you as we move along, but you remember here that motor unit, yes, is going to be a motor neuron which will actually innervate a certain group of muscles, so to speak. So we are going to look at those things. We are also going to look at the composition of the muscle. We're going to look at all these ones as we move along. And then one thing I have to tell you is that when you look at the skin, deep to the skin or just below the skin, we're going to have what we call the hypodermis, and therefore the hypodermis is what we call the superficial fascia. So if you hear of superficial fascia, that's the layer, yes, usually of hypodermis, subcutaneous tissue beneath the skin. And of course, we also are going to have deeper, for connective tissue that we call it deep fascia. Now remember, the word fascia has to do with covering, so to speak. So there will be another deep fascia. Now the deep fascia usually they will cover these muscles. So we're going to see that yes, we're going to have muscles will have deep fascia which will cover them. So that is one thing that we are going to see. All right, so moving on with these myology thing that we are doing, I just want us to actually understand that yes, muscle contraction, generally if muscle contracts, it causes shortening of the muscle fibers, which actually generate that kind of movement that we are looking at. But mainly for purpose of studies, we are going to see that there are three kinds of muscle contraction. And these three kinds of muscle contractions are grouped into two main groups or two main categories. And these two main categories, yes, are what we call the isotonic contraction, and of course isometric contraction. So the one you see here, this isotonic, there's also isotonic. So the word iso means same, and tonicity has to do with tension. So it means the same tension, okay, is generated. So this represents what we call the isotonic contraction. And then there's another one which is the isometric contraction. And iso means the same, and metric has to do with measurement, so the same length, so to speak. So let's look at yes, with respect to movement of the either flexion occurring or extension occurring at the elbow joint. Now this muscle over here that we see is the biceps brachii muscle. Now if we talk about isotonic contractions, there are two kinds. We have this first one known as concentric contraction. Now in concentric contraction, yes, what will happen is that yes, for instance, in this case, concentric contraction is actually the classical movement contraction of muscle, so that upon contraction there's the same developed tension is there, but the muscle shortens, so the muscle fiber shortens. So you can see that this one, the fleshy muscle over here that you can see, is a bit elongated compared to this one, so this one has shortened. So for instance, if you are raising an object, okay, let's say an object that you can easily raise, for instance this dumbbell, raising it, okay, then what will happen is that this biceps brachii muscle will shorten, and that is what we call concentric contraction, and it's a form of isotonic contraction. Now the other one, where of course, for instance, so in this case, if we are moving the object upwards, okay, then that's what we see. But in situations where, let's say, you are trying to try to lower down, okay, or during the downward phase, actually lower down the object, then what will happen is that you see what we call eccentric contraction. Now for eccentric contraction, rather, now look at the belly of this muscle. This one is shorter, but this one is longer. This one, the same developed tension will be there, but what will happen is that the muscle fibers will actually elongate. So in eccentric contraction, which is opposite to concentric contraction, the muscle fibers elongate. So that is what you see in eccentric contraction. So that eventually what I want to explain is that with respect to the elbow, then what will happen is that if it's concentric contraction, then it will lead to flexion of the elbow. So we can see that the angle over here has been reduced. Okay, this one is greater compared to this. But in eccentric contraction, then what will happen is that it leads to what we call extension, so that the angle actually increases. All right, now the next one is the isometric contraction. Now for isometric contraction, yes, the length remains the same, but the tension increases. So for instance, if we are moving something, but one thing over here is that the muscle fiber does not change, as we said. So isometric, the length remains the same. So that, for instance, you want to move an object, okay, a stationary object, a very object that you can't move it. You try everything, it doesn't move. Then it means that those muscles that are supposed to cause the movement will be in a state of isometric contraction. Again, for instance, this dumbbell is neither moved up nor moved down, and therefore it stays the same. And that is what we call isometric contraction. So that is one thing that we see. So it means that in isometric contraction, neither is flexion nor extension taking place. All right, now let's look at some bits regarding how muscles are named. So regarding the nomenclature of muscles. Now this one is respect to the shape of the muscles. Now let's look at this muscle. For example, this is the biceps brachii muscle, which we take a cross section at the muscle belly. Now these represent the fascicles. Now I'm going to explain the fascicles. Now fascicles represent bundles of muscle fibers. So you can see that the muscles of bundle fibers are moving parallel to one another, And once we see the fleshy muscles moving parallel to one another, then we see that the muscle, the shape is actually showing what we call parallel arrangement. Now, this one too is showing parallel arrangement. Now, in the situations where for the rectus abdominis muscle is showing parallel arrangement. Only that we can see these whitish bands over here, and that is what we call tendinous intersections. So that the abdomen, the muscles over there, will give us this kind of six-pack arrangement. But of course, this one is going to have more than even the six-pack arrangement. Okay, 2, 3, yeah. So it's going to have that kind of six-pack arrangement. Some people may even have that kind of 8-pack arrangement. So what we want to show here is that the muscles fibers, or the muscle fascicles, which I'm going to explain pretty soon, are running parallel to one another. Then this one showing the wrap, having that kind of wrapping arrangement, and therefore this one is seen in situation of the spinator muscle. Yes, these muscles, yes, they are not really moving. The fascicles are not moving. Parallel. Yeah, they are moving somewhat parallel here, but at the end of the day, they come and converge over here at the point where they eventually form a tendon over here, and therefore we call this arrangement convergent arrangement. Now we have some muscle fibers which are feather-like, like the feather of a fowl. Yes, feather-like. Now some of the feathers, depending on how they are, yes, this one is having just one feather, one particular direction, and therefore we say it is unipinnate arrangement. And of course we see this unipinnate arrangement in extensor digitorum, yes, muscle. You can see that one there. Yes, even apart from that, there's a lateral two lumbricals, L1 and L2 lumbricals are also unipinnate. If you look at the palmar interossei, they are also unipinnate. Of course, even bipinnate. Now you can see that the muscle fiber, they are running in different directions, so feather-like, but taking two different directions. And you can see example in rectus femoris, muscle is there. Yes, even the dorsal interossei muscle. Yes, and then of course the medial two lumbricals, L3, L4 lumbricals, they are also going to be bipinnate. Yes, this pectoralis muscle is an example of the convergent type. Yes, for multipinnate, we are going to see it in this muscle, which we call it the deltoid muscle. The deltoid muscle is multipinnate. Yes, some muscle fibers, yes, the fascicles or the fibers actually have this kind of circular profile. Now when we see that, then we see it is orbicularis, having this circular profile. So for instance, orbicularis, oris or orbicularis oculi muscles, yes, are going to have this circular profile. So that is one thing that we are going to find actually. Now, with respect to continue with the nomenclature, yes, we can have a situation where, depending on where the muscle is located, for instance, talking about tibialis, anterior muscle, Yes, because it's located, yes, anterior to the tibia. So we call it, tibialis, anterior muscle. Yeah, we call it tibialis posterior muscle will also be there. Yes, if you look at this, muscle over here, the occipital, frontalis muscle. This is a frontal bone because of its location, and there's occipital bone over here, and then for the muscle over there will be occipital frontalis muscle. Now look at this. This is an aponeurosis. Now we said that aponeurosis will connect muscles, which will connect skeletal muscles. Aponeurosis. Then we say that if it's a tendon, then it will connect, yes, muscle to bone. So there's a tendon connecting muscle to bone. Now remember that this aponeurosis is even, a tendon, but it's a broad, flat, glycerin tendon. Ligament, of course, we said that will connect bone to bone. Bone to bone will be ligament. Now, this, look at this. Look at the shape of this. Having this saw-like shape appearance, as if there's a saw, the tooth of a saw-like appearance, saw-tooth shaped appearance. And this is what we call serratus. So once you see serratus, so of course serratus anterior muscle, yes, serratus posterior superior, serratus posterior inferior. Muscles depend on the shape, but mainly this one I want to show the location. Then there are some muscles which, of course, we call some of them to be, maximus. So if you hear based on size, maximus, then it means it is large. Minimus means, it is small. Longus means that's the longest. Yes, brevis means short. Yes, even when you look at, so brevis is short. Yes, brevis, is short. If you look at major, yes, for instance, pectoralis major, yes, means, major means, large, and then minor means, small. And then, of course, for instance, gluteus maximus, yes, gluteus minimus, even gluteus medius, then it's intermediate between these two. That's in terms of size. Longus capitis, yes, abductor pollicis brevis, yes, all these are. So you hear on the word brevis, then it's short, then longus are the long form, and then major means, of course, large, and then minor means small. So for instance, pectoralis major, pectoralis minor, teres major, teres minor, are some, examples that we can give. Of course, psoas, minor muscle, psoas major muscle. I can see that the psoas minor muscle is small compared to the psoas major muscle over there. So that is one thing that we are going to find, with these ones. All right. Now, in terms of how the muscle fibers, yes, are moving, we can also name the muscles. So for instance, in the abdomen, especially we can have, the rectus abdominis muscle running in straight manner. Yes, you can talk about the rectus, femoris muscles over there will also be examples. With the muscle fibers are running, actually parallel to the long axis of the body. Then, of course, if it is oblique, then it's neither going through, the sagittal plane nor the coronal plane, nor, yes, even the transverse plane, but running in a different manner, obliquely. And so we're talking about, the external oblique, internal oblique muscles. And then, of course, some of the muscles will run, in a transverse manner. The fibers will run in a transverse manner, which will be actually perpendicular to what we call the long axis of the body. And that is what we call, transverse. So for instance, the transversus abdominis muscle is an example in this regard. Then we can name, muscles based on the number of hairs or the number of origins. Okay, that is has. Now, if it is having actually two origins and for that matter two hairs, then we call it biceps. So for instance, we have the biceps brachii muscle over here. Okay, so that one of its heads, yes, it's coming from, the coracoid process, and then one of its head, that is actually the short head coming from the coracoid brachialis, coracoid process, of the scapula, and of course the long head will actually come from the supraglenoid supraglenoid tubercle, and therefore we will say it is a biceps. Now because it's located around the arm area and the brachium is the arm, then we call it biceps brachii muscle. Then we can also have triceps. Again, triceps here in the posterior compartment of the arm. The triceps muscle is there because having three origins or three hairs. Then you can also have, if it's having four hairs or four origins, that like quadriceps, femoris, muscle. So it's an example of the quadriceps, group. Now we can also name muscles based on their origin, and insertion, So for instance, we have this muscle known as sternocleidomastoid muscle. The sternocleidomastoid muscle is having attachment to the sternum. So having two hairs actually, one from the sternum, one from the the medial end of the clavicle, and then inserting on the mastoid process, and therefore we call it sternocleidomastoid muscle. Yes, talk about the stenohyoid muscle, the stenothyroid muscle. Yes, all these are examples based on the attachments that the muscle has. Yes, we can also name muscles based on the kind of action or the function performed by the muscle. So for instance, we can have flexors, we can have extensors, we can have adductors, we can have abductors, we can have supinators, we can have pronators. So these are how we can name muscles. So for instance, we can have flexor carpi radialis muscle, we can have extensor carpi radialis longus and brevis, because they are going to actually cause either extension or flexion at the wrist. For instance, abductor pollicis brevis, the word is abductor. So the word is that is going to actually cause, this is a pollicis, has to do with the thumb, move the thumb away from the rest of the digates. Then there's adductor pollicis, adductor pollicis muscle, then it's going to add the thumb to the other digates. And of course abductor magnus, abductor is going to cause so for instance this adductor magnus muscle, which is going to cause the thigh closer to the midline of the body, bring the thigh closer to the midline of the body, adduct. That is abductor in that manner. Then you're also going to have what you call extensor digitorum, it's going to cause extension of the digates. So that's how we can name muscles based on the function that they are going to perform. Now other things that I want to show you is actually the location. With the location again, even the gluteus, we talk about gluteus, and it has to do with the buttocks area. And of course, for instance, we're going to have gluteus minimus, gluteus maximus, gluteus medius. Then if it is pectoralis, then it has to do with the chest area, the thorax, pectoralis. So pectoralis major, pectoralis minor. Then if you're having muscle having a certain number of fleshy bellies, for instance, if the digastric muscle, digastric muscle is going to have two bellies, so giving it that name. Then based on the structure, so for instance semimembranosus is quite membranous, then we're going to have also semitendinosus, quite more tendinous actually. Then based on relative position too, we can actually define muscles or name muscles. Of course, that's medial and lateral pterygoid muscles, which are actually mastigatory muscles. So these are actually examples that we can actually have. So that is how we are going to name muscles. Now, other concept that I have to explain is that a muscle will have an origin and an insertion. We'll actually have two attachments, an origin and an insertion. Now the origin is usually immovable, and then the insertion is the movable part, and therefore the action of the muscle will actually take place at the level of insertion. So that is one thing that we actually have to understand regarding these muscles. I mean, regarding the attachment, we have an attachment. One of them is the origin, where it originates, immovable, and then the point of insertion, which is movable. Yes, apart from that, we're also going to have what you call the prime mover, or the prime, or the agonist. So this prime mover, yes, will be the muscle which will actually going to produce the movement of interest. Okay, so for instance, if it's extension, then that is the agonist, or the prime mover, the movement that we are actually interested in. Then, apart from that, we're also going to have the antagonist. So for instance, let's talk about the external intercostal muscle. So for instance, if external intercostal muscle is going to contract to cause movement of the rib cage, upward movement of the rib cage, then of course the internal intercostal is going to cause relaxation, to cause the opposite to happen, and therefore you can see the action of interest. If we are working against the action of interest, then we say you are an antagonist to the prime mover or the agonist. Then we're also going to have some fixers. There will be some muscles which will actually help stabilize or actually potentiate the action of the prime mover. We call them fixers. Yes, apart from that, we're also going to have some muscles that we call them intrinsic muscles. We're going to have others that we call them extrinsic muscles. Now, if an intrinsic muscle, then we are within the organ or the structure of interest which we are going to work there. So for instance, we can talk about the intrinsic muscles of the tongue. Of course, the longitudinal muscles in the transverse muscles in the tongue. Then we can also have the extrinsic muscles of the tongue, which are not actually going to find themselves within the tongue, but they are going to work to cause movement in the tongue, and therefore they are called extrinsic muscles of the tongue. Extrinsic muscle. And the extrinsic muscles, yes, for example, with respect to the tongue, then we can talk about the hyoglossus muscle, because it's not actually part of the tongue. It's not a content of the tongue, but it's actually going to cause some movement of the tongue. Now, usually, if it's an intrinsic muscle, they will actually cause what we call fine movement. Actually mainly changing the shape, but extrinsic muscles will cause movement, which will actually mainly changing actually the position of the structure. All right, so let's look at actually the organization of the muscle. So from the gross aspects, for example, there's the bone, and then we have this muscle. So connecting the bone to the muscle is the tendon. Now the tendon is made of dense regular connective tissue. And then over there now you can see this is the bigger muscle that we are talking about, the bigger picture muscle we are talking about. For instance, if you get some meat, let's say chicken, and you find it that you see some whitish substance on it, which of course you can remove. That is the connective tissue that surrounds the muscle, the bigger muscle that we are talking about. And that is what we call epimysium. Epi means above, epimysium. Epimysium is made of dense, irregular connective tissue. Epimysium, dense, irregular connective tissue. All right, now this epimysium, as I said, is going to surround this whole muscle, or let me say muscle as an organ. This whole muscle will be surrounded, bigger muscle, by epimysium. Now, assuming you take a cross section of the meat you are going to take, then you may see that there may be some areas over there as if there are some strands in there. Those ones that you see are what you call fascicle. So this is what you see, fascicle, which we actually magnified one of them. This is the fascicle. Now this fascicle is also surrounded by a layer of dense irregular connective tissue. Remember, dense regular connective tissue, epimysium surrounding that whole big muscle, is dense irregular connective tissue. Now, if you take one strand of it, and that's a bundle of muscle fibers, that's what we call fascicle. Fascicle. So fascicle is made up of these units known as muscle fiber. So several units of these muscle fibers are coming together, will be wrapped in this connective tissue, dense irregular connective tissue known as perimysium. Peri means around, perimysium, forming this fascicle. Now, if you look within the fascicle, what happens is that, yes, I said that individual muscle fibers are going to come together to form this fascicle. Within it, we have some whitish areas also in there, as you can see, and that is what we call endomysium. Endomysium. Endo means within. Endomysium, within the fascicle. And that is what, it's made of loose connective tissue, actually areolar connective tissue. And this one is rich in blood vessels, which will actually nourish, the muscle fibers. Now, if we talk about muscle fiber is actually the cell of the muscle. The basic unit of the muscle is known as the muscle fiber. Therefore, you can also call the muscle fiber muscle cell or myocyte. So that is the muscle cell. Now, within this muscle cell, you can see some other strands, okay? And you can see this cross-section area. And this over here is known as, each of these piece of strands that you find over there is known as myofibril. Okay, that's what you find over here, myofibril. Then beyond the myofibril, this myofibril will be made up of what we call myofilament. Myofibrils will be made up of myofibral filament, which we are going to look at. So, that's one thing that I want you to actually appreciate, that from the gross level, tendon will attach the whole big muscle to the bone, covering the entire muscle will be epimysium, dense irregular connective tissue. If you take a strand out of it, yes, the one you can see is known as fascicle. Fascicle consists of microscopic structures which are known as myocytes, or muscle fiber, or muscle cell. Now, if you take the muscle cell, it's made up of myofibril, and within the myofibrils are made up of what we call myofilaments, which we are going to actually see. Now, there are some terms that I want to actually explain. Now, if we look at the muscle cell, the muscle cell will actually have a membrane. So the membrane of the muscle cell, instead of calling it plasmalemma, or of course cell membrane or plasma membrane, we will call it sarcolemma. Or we said that sarco has to do with muscle, sarcolemma. Yes, apart from sarcolemma, yes, the cytoplasm of the muscle fiber or the muscle cell is known as sarcoplasm. Apart from this, yes, the smooth endoplasmic reticulum of the muscle cell is known as sarcoplasmic reticulum. Then, of course, if we talk about the contractile unit, which I'm going to show to you, which is found between two Z lines, which I will show to you, the functional and contractile unit of the muscle is known as sarcolemma. Now, one key thing that I have to explain to you here is that the muscle cell, like, if you look at the blood will have, the red blood cell will actually have what we call hemoglobin. The muscle cell will be endowed with myoglobin instead. And within the muscle cell, yes, now we know that glycogen will be stored by the liver cells. But in addition to the liver cells, we also have what we call the muscle cell, which will also store glycogen. But unlike the liver cells, which of course will release the glycogen as glucose into systemic circulation in hypoglycemic states so that the rest of the cells will enjoy it, myocytes or muscle fibers, they don't do that. They use the stored glycogen for themselves alone. Okay, so they also store glycogen. So far, yes, the vagina will store glycogen. The liver cells will store glycogen. The skeletal, the muscle cell actually will store glycogen. Okay, will store glycogen. So that is one thing that we have to understand. Now, I said that within this muscle cell will be myofibril. Myofibril, as you can see, is taken out over here, which we have actually enlarged over here. Now, if you look at this, we have some key concepts that we have to explain. I said that within the myofibril will be what we call myofilament. So actually, this myofibril, is actually a bundle of protein myofilaments, and these protein myofilaments are those ones that I'm going to explain to you now. Now, if you take the myofilaments, okay, we have what we call the thick filaments and the thin filaments. So as you can see, there are some areas which are a bit thicker. So you can see that this one, it's quite deeper. So we say thick filament. We have the less dense ones representing the thin filament. So we have thick filaments. Now remember that thick filament, the diameter you can see is larger than that of the thin filament. So there's a thin filament, there's thick filament. Now the thick filaments will have a diameter of roughly 11 nanometers. So that's 11 times 10 to the power minus 9 of a meter. So 11 nanometers. And the thin filaments will be around 5 to 6 nanometers. 5 to 6 nanometers. Now, the thick filaments are mainly made up of what we call myosin molecules. Thick filaments are mainly made up of myosin molecules. And then thin filaments are made up of mainly what we call actin, together with the regulatory proteins, namely tropomyosin as well as troponin. So that is how they are. Now, one other thing too that I have to make mention of is that there are other proteins, the other filaments, so to speak, apart from the thick filament and the thin filament. There are also what we call elastic filaments, and that one we don't see it over here, but they actually measure roughly 1 nanometer. That's with the elastic filaments. And these elastic filaments constitute of mainly titin as well as of course connectin. That is what we find. Now, what these elastic filaments do is that they keep the thin and thick filaments aligned over each other for proper or effective contraction. Now, these elastic filaments will also be important in forming this zigzag shaped structure that you find over here known as Z-zix or Z line or Z line. Okay, so for instance, titin will contribute, which is an elastic filament, will help in the formation of the Z line, which I will be coming to pretty soon. Now, having seen the thick filaments made up of mainly myosin. Now, myosin will have actually 2 heads and a tail, globular head and a tail. Now, actin filament. Now, for actin, we have what we call F actin and of course G actin. Actually, the G actin is a subunit of the F actin, so that the myosin head, that's the active site for binding by the myosin head. So myosin head will bind to actually F actin through what we call G actin. G actin. So let's look at this. Now, if you look at these ones, okay, if you look at the A band, now one thing is that yes, these myofilaments will arrange themselves in different ways to produce various shades of colors under light microscopy, which we'll be seeing pretty soon. Now, what you see over here is that, look at this, from here to here represent what we call the A band. A band. A for anisotropic, so A band, anisotropic band. Now this A band, yes, is formed by parallel arrangement of the thick filaments, okay, as you can see, thick filaments, and of course they overlie the thin filaments. So actually the A band will mainly contain thick filament as well as of course thin filaments. Thin filaments will make up this A band. Then apart from this, so it tells us that the A band mainly will contain a lot of myosin as well as some bit of actin as well. Now if you move this way or this way, we have what we call the I band. Now the I band is the isotropic band. And for the I band, yes, we are going to find only actin filaments. You can't see any thick filament over there. So you find only actin filaments. Okay, actin filaments. So that is what is going to form the I band. I band. Now look at this. Now within, actually dividing, okay, so we can see this disc within the I band. So within the I band is this disc, as I said, made of actually elastic filaments, mainly tightly. Okay, this is going to be help actually connect, serve as the means so that we can have what we call the functional unit of the muscle fiber. So from one Z line to another Z line, adjacent ones, we have what we call the contractile unit of the myocytes. Okay, and that is what we call sarcomere. So the Z line will actually appear, okay, at the middle point of the I band. Okay, middle point of the I. So this I band, okay, dividing it into two. Okay, essentially that's what it's going to do. We're going to bisect the I band, and then for adjacent ones we are going to have what we call the sarcomere, the contractile functional units of the muscle cell. Yes, apart from this, now you will also find that within this A band, we can also find the M line. So within the A band, you can see this M line. Now this M line, which actually runs this way, as you can see, it's a bit thicker. What it does is that it anchors the adjacent thick filament. So the adjacent thick filament will be anchored by what you call the M line. And it's actually within the middle portion of what we call the A band. Then there's this light area over here, which is divided by actually the M line. This light area is known as the H band. Okay, so the H band is this middle lighter region over here within the A band, okay, that contains thick filaments. Okay, so you can see there will be only thick filament over there. That is when the muscle fiber hasn't contracted. But of course, after contraction, what will happen is that the thin filaments, yes, they may actually get closer over here. But remember that in any case, thin filaments don't move actually upon contraction. It's actually the Z lines which will actually get closer upon contraction. So this area, the H zone, is this light area of the A band that you find over there. Now, one other thing too that I want to actually show you is what we call T-tubule. T-tubule. Now, I explained that the cell membrane of the muscle cell is known as sarcolemma. Now, the sarcolemma, okay, on the surface will invaginate, will move deeper into the muscle, okay, internal aspect of the muscle cell to form what we call transverse tubules. So they run transversely into the interior of the myocyte, forming this tube-like, channels. Okay, forming these tube-like, channels, and this is very important so that we can convey electrical impulses on the surface of the myocyte into the deeper portions of the muscle cell. So that is one thing that we can actually find. Now, associated with the muscle cell, that is actually a skeletal muscle cell on either side. Now, I've also explained that yes, there will be sarcoplasmic reticulum, which will be very important for storage of calcium. Now, remember that calcium is very important when it comes to contraction of the muscle cell. Now, there are dilatations which are associated to what we call the T-tubules on either side of the T-tubules. Okay, so this way, so you can see there's a cystine, terminal cystine, the end point. Okay, terminal means end, and cystine has to do a dilatation or sac-like structure over here, and there's another cystine over here. Or this is what you see. There's a cystine over here, and in between them is the transverse tubule, together forming what we call triad. So actually in a skeletal muscle cell, we are going to find triad, but in cardiac muscle, we are going to find dyad. But we don't find this system in smooth muscle. You don't find this system in smooth muscle. So essentially, this is what we have to actually understand, regarding the myocyte organization, as well as together with its molecular aspect. So that is what we actually saw. Okay, this is what we actually see. Okay, these ones representing the connecting as well as tighten are a bit larger compared to connecting filaments, which are both elastic filaments. Then of course thick filaments, yes, mainly by what we call the myosin. Then of course thin filaments by what we call actin filament, together with the regulatory proteins. So we explained these ones. So as I said, myosin will have hairs. Each myosin molecule will have a head, will have two hairs, and then of course a tail. Then these are the actin, the active site where so actually F actin, and the active site where the globular hairs of the myosin will attach during muscle contraction. The sliding filament method will be by what we call the G actin. So these are actually the regulatory proteomyosin, and of course protonin troponin complex molecules over there. So as I indicated, we have what we call motor units. Now you can see this is a muscle cell, there's another muscle cell, this is another, but they are all supplied by this motor neuron. Therefore, one motor neuron together with muscle fibers, it supplies, forms what you call motor unit. So that is one thing I want to show you. Therefore, it tells us that yes, there will be that kind of connection, because between a motor neuron and a muscle cell, then we're going to form what we call neuromuscular junction. Therefore, neuromuscular junction will contain the what we call motor end plate, and this motor end plate will be that kind of muscle skeletal will be related to the muscle. So we expect that there will be a motor end plate over here because this is the muscle cell, and then you want to have what you call the terminal part of the muscle known as the synaptic knob. Yes, this synaptic knob can also be referred to as Bowtons or Bowton, actually Bowton synaptic knob, also known as Bowton. So at the molecular aspect, yes, at the neuromuscular junction, we are going to find the motor end plate, and then we're also going to find the synaptic knob or the Bowton. And then in between them, there will be a potential space over there known as synaptic cleft. So what happens is that, yes, there will be vesicles, okay, within over there, which are, contain neurotransmitters, for example, acetylcholine, and then of course, by way of docking with the membrane of the motor end plate, okay, of the muscle cell, then there will be release of those neurotransmitters, and then there will be receptors by what we call, so these are the receptors by what we call the muscle cell, so that they can actually cause change in the action potential, membrane potential of the muscle cell to cause various impulse generation. So that is one thing that we are going to find over here. Now, with respect to muscle tissue, I've indicated that there are three main kinds of tissue that you're going to find. You're going to find, of course, the skeletal muscle. You're also going to find, yes, the cardiac muscle, and we're going to find the, smooth muscle. Now, what will be the identifying, yes, features which will help us distinguish between these muscle types? So this is actually the transverse section of the skeletal muscle tissue. This is actually a longitudinal section of it. Now, what we find is that, you see, at the transverse section, you can see that it is having this rounded ends. So the profiles, giving us roughly this kind of rounded, ends. All right. Now, with respect to the characteristics of skeletal muscle cells, we say that, yes, the shape, roughly, cylindrical shape, as you can see in this longitudinal section. Yeah, cylindrical shape. The fibers run in cylinder. They are in cylinder, so to speak. And then they are actually, multinucleated. They are multinucleated. So there's one nucleus over there's one nucleus. Now, if you look at this muscle cell, this myocyte, you can see having 1, 2, 3, 4, so several nuclei. So therefore, we can see that skeletal muscle cells are multinucleated, and therefore structurally form what we call syncytium, multinucleated cell syncytium. Now, apart from this, yes, the nuclei are actually peripherally placed. Okay, they are not placed in the center of the muscle fibers, but they are placed at the side. Okay, peripherally placed nuclei. Then, apart from, yes, their peripheral nature of the nuclei, it's an important thing that we have to know. Now you can see that some kind of light and dark bands that you can see, and that is what we see under light microscopy. That's the A and I bands that you can see forming this kind of cross striations, and you can actually also appreciate it here. Striations. So once you can see striations, then it tells us that, yes, it's likely going to be skeletal muscle. But of course, there's another muscle which will have striations, that's the cardiac muscle. So this is its cross striations. Now you can see that the fibers do not branch. Mainly, the fibers are not going to branch, but there could be some areas where skeletal muscle fibers may branch. But generally, skeletal muscle fibers won't branch. Now, one key thing that we have to understand, yes, is that even they are actually voluntary, voluntary by way of function, because yes, they will be innervated by they will have innervation not coming from autonomous, but from actually somatic nerves, so that they are actually, yes, voluntary in nature. But they can easily get fatigued. You can easily get fatigued, and they go through graded 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 contraction. So that is what you find with the skeletal muscle. Now the skeletal muscle is usually associated with, of course, bones of the body. Okay, so talk about the various muscles. Yes, we have in the head and neck area, in the limbs, yes, and everywhere in the abdomen. You know, will be abdominal wall, pelvic wall. These muscles, yes, will be, yes, of course, skeletal muscle, skeletal muscle. All right, so identifying features. Cylindrical shape fibers. They are going to have, they will be multinucleated peripheral place nuclei. They will be cross striations. Fibers don't show branching. Of course, they have rounded ends. Then, by way of function, they are voluntary. Then, of course, they can easily get fatigued. They go through graded contraction, and they are actually associated with bones. Now, having talked about this, now there are three kinds of skeletal muscle fibers. There are three kinds of skeletal muscle fibers, and these are what we call the red or slow fibers. Also have of course the white fibers, okay, or that is the fast glycolytic, and I'm also going to have the intermediate fibers, or the fast oxidative fibers, which we are going to see them. So if we look at these ones, okay, the red ones, they actually appear quite red because they contain a lot of myoglobin, and they are actually more highly vascularized. Actually highly vascularized. Now, because they contain a lot of mitochondria, then they go through actually aerobic respiration. Okay, so that they go through aerobic glycolysis, and of course go through the Krebs cycle, and of course oxidative phosphorylation to generate a lot of energy in the form of ATPs. Now what happens is that they usually have smaller diameter, relatively. They have relatively smaller diameter. Now because they go through aerobic glycolysis, then they usually don't go through fatigue. They are slow to get fatigued. They are slow to get fatigue. And of course they can actually produce low power contractions over a long period of time. Low contractions over long periods of time. Now remember that whatever muscles we have in the body will have these kinds of muscle fibers, but there could be areas where you have predominance of a particular fiber than the other. Now with this kind of red fibers or the slow oxidative fibers, which are staining very dark over here, they are going to find themselves mainly in the back muscles. In the back muscles, you are going to find them there. For instance, latissimus mos, dorsi, yes, you are going to find them there. Then we are also going to find these ones, which are the intermediate groups. Okay, the intermediate groups, which you find over here. Okay, so the intermediate groups, okay, groups over here that you find. All right, so those ones are known as fast or the intermediate oxidative group. Okay, so these ones, yes, they will contain although lower than that of the slow oxidative, they will contain what we call the relatively, yes, less myoglobin, and of course less blood vessels compared to that of the slow oxidative one that we've seen. Then of course they will go through actually mainly aerobic respiration. Therefore, but sometimes they may actually go through anaerobic glycolysis to generate energy when the demand for energy is high. But one thing is that, yes, if they go through anaerobic respiration, they can easily become fatigued. They can easily become fatigued. Therefore, they can easily get fatigue compared to that of the slow oxidative group. Then mainly where we are going to find them will be in the limbs, muscles of the leg, lower limb, that of the upper limb. We are going to find these ones there. We're going to find this intermediate group, fast oxidative group. And they may, switch, to anaerobic, as we've said, and then they could easily become fatigued. Have to actually get to that one, get to know that, because yes, mainly they will go through aerobic glycolysis and of course go through oxidative phosphorylation, coupled with Krebs cycle. But what happens is that yes, if the demand is too great, then they will switch to anaerobic glycolysis. Now the ones that we've indicated W, yes, represent actually the fast glycolytic fibers, and they actually pale, so they don't really pick up the steam, and they have the fastest contractions, and they are actually going to go through anaerobic glycolysis. So they generate a lot of lactic acid, which will actually cause burning sensations within it. Therefore, they easily get fatigued than the other muscle fibers that we find. So that is one thing that we find, right? Now this is the cardiac muscle tissue. Now for the cardiac muscle tissue, what we will find is that they will have roughly is somewhat rectangular shaped fibers, but they show branching. As you can see, they show branching. These are the muscle fibers, they show branching, and they are actually mononucleated. They are actually mononucleated or binucleated or mononucleated. And one thing is that the nuclei are located at the center, centrally placed nuclei. Now one key thing is that yes, they also find these cross striations. Therefore, you don't find cross striations exclusive to skeletal muscle. You also find it in cardiac muscle, but the branching is quite unique to them. They show this peculiar branching. Again, as you can see, there are some darker regions over here, which represent what we call intercalated disc. Now these intercalated discs allow the muscle cells, which we call them cardiomyocytes, to actually contract in unison. So that what will happen is that there will be that kind of electrical impulse spreading to various cardiac muscle, so that they contract in a simultaneously in a coordinated manner. Therefore, because of these kind of intercalated discs, we say cardiac muscle cells form what we call functional syncytium. Their ability to contract in unison in a coordinated manner make them to be regarded as having what we call functional syncytium, exhibiting functional syncytium. Now one thing that we have to actually say is that yes, these ones, they contract actually very fast. Although they contract very fast throughout life, yes, you are going to find it associated with the heart, so it's going to contract very fast, and it is non-fatigable, surprisingly. So it means that it goes through only aerobic glycolysis, and of course Krebs cycle coupled with oxidative phosphorylation to generate energy for it. So that is one thing. Yes, one thing is that yes, this one doesn't depend on stimulation from external source by way of nerve stimulation, because it's myogenic. It doesn't rely on any nerve stimulation. It is having this auto-rhythmicity that I can contract and relax on its own. Now these gap junction, these intercalated disc, yes, therefore will serve as communicating channels, okay, between the muscle fibers, and therefore you can see they represent what we call gap junctions, and they also contain actually desmosomes. Now for the areas where you find the cardiac muscles, we find it, yes, associated with the, I mean, the myocardium, yes, of the heart. Apart from that, the great, vessels that are associated with the heart, okay, either they are terminal portions or the proximal portions, okay, will be actually having some bit of cardiac muscle cells. For instance, talking about we talk about uh, the proximal aspects of the pulmonary trunk, talking about the proximal aspects of the as ascending aorta, talking about the distalmost aspect of the superior and inferior vena cava. They are actually going to have what you call cardiac myocytes, some bit of cardiac myocytes, although mainly they will have what you call smooth muscles. So that is one thing that we're going to see. Now this is smooth muscle. This is smooth muscle. Now for smooth muscle, as you can see, their shape, you're going to have this spindle shaped appearance, okay, with their muscle cells tapering at both ends, okay. And one thing is that they are mononucleated. They have a single nucleus, and then the nucleus is located at the center of the cell, so centrally placed nuclei. They don't have any cross striations. We don't see cross striations, okay. So this is the only muscle cell without cross striation. It doesn't show branching, and then one thing too is that, yes, it's involuntary, okay. So because we're gonna supply by to the most nervous system having parasympathetic and sympathetic innervations. Now one thing that we saw, which I'll go to, which when I get there I'll explain it to you. There are no T tubules. I will explain that one to you in the next slide. Now what happens is that, yes, these smooth muscles will have several gap junctions. Smooth muscles will be endowed with a lot of several gap junctions, so that there will be cell-cell communication, cell-cell communication. Now with respect to the areas where we will find this smooth muscle fibers, yes, we are going to find it, yes, in hollow organs, internal hollow organs mainly. Yes, if you look at the GIT, yes, the muscles over there mainly the muscularis mucosae, yes, from the middle one third, lower one third of esophagus, yes, the entire stomach, small intestine, yes, large intestine, they will have smooth muscles. Yes, even the respiratory tract, yes, the bronchioles, the smooth muscle of the bronchioles will be actually smooth muscles. Then even the respiratory system, yes, even the urinary tract, yes, you are going to find mainly the tubular structures to be mainly smooth muscles. But one thing I want to mention is the erector pili muscle. The erector pili muscle, yes, although it's associated with the skin, it is made of smooth muscle. It is made of smooth muscle. Talk about the sphincter pupillae, dilator pupillae. Yes, they are also made of smooth muscles, smooth muscles. Now, one thing is that, yes, we've described a general organization of muscles, but smooth muscles don't usually follow the trend of skeletal muscle or cardiac muscle, because what happens is that smooth muscles do not have T tubules. They lack T tubules that we saw, but rather they have these things. Now I said that T tubules will actually penetrate the inner portion from the modifications of the cell membrane, okay, sarcolemma, into what we call the interior of the muscle, okay. But smooth muscles do not have it. They rather have what we call caveoli. So these several openings over here, okay, will serve the purpose, okay, will be analogous to what we call the T tubules. So smooth muscles lack T tubules. Apart from that, they also lack what we call the myofibrils that we saw, and therefore they don't have any Z line, and therefore they don't have sarcomere. They don't attach to tendons anyway. together, and they don't form, aponeurysm, are not attracted to, any aponeurosis as well. But one thing is that we also mentioned that one of the regulatory proteins was troponin, helping in muscle contraction. Smooth muscles lack, this troponin, but rather they have what we call calmodulin. Calmodulin. So calmodulin is, there to help them in muscle contraction, smooth muscles to contract effectively. Again, even the area for calcium storage, which is the sarcoplasmic reticulum, is even less developed because, of course, it has what we call calmodulin. Ladies and gentlemen, this has been, yes, a very long tutorial. I hope you find it helpful. May God richly bless you.